Welcome
to
Emad and Phillips.
Biolab World.
Alright, so today what are we studying, Professor Phillip?
We are studying mitosis and meiosis.
And these are the processes of cell division. Mitosis is regular cell division, where one cell divides into two cells. And it's with a regular cell, like skin cells, or ear skin cells, or eye cells. There are no really eye cells.
No cells.
But no cells, lip cells.
Any kind of cells.
Any kind of cells that are not sex cells. Meiosis is the division of sex cells. And today's experiment is looking at mitosis and meiosis. What we're doing is we've got a set of HeLa cells which comes from Henrietta Lacks, a woman that died in 1951 from cervical cancer. Her cells have continued to divide because there's cancer cells that never have like a stopping stage where they're stopped from cell division.
Scientists have been using them for years, and they're still using them today. And these are what we're using. And we're gonna be taking some awesome footage with these eye touches. Cheese. Brought to you by Dr. Weber.
Let's get started.
Hey guys.
Welcome back. So, what we've been doing is looking at these HeLa cells that we were talking about before through a microscope. We will include some of the pictures that we took in the video later. That's just a preview.
Chromosomes.
See, what we did is we dropped the medium that they were in, that the cells were in, from about 8 inches above a slide. We dropped them onto the slide.
HeLa cells.
It's in that container thing.
Kept in ice.
And it smelled like vinegar.
Yes.
At least to us. So, what we did is we dropped them from a pipette onto a slide so that the cells would burst and we could see their chromosomes.
Now we laid our slide flat.
And yeah.
And
when they dropped, we let them dry. We stained them and we put them on the microscope.
And the purpose of dropping them from 8 inches above is so that when it hits the slide, the cell will burst open and the chromosomes will become. They'll come out in the environment. That way we'll be able to view them.
And once again, we'll include these pictures later on in the video. Back. So Professor Emad here talking to you about mitosis. Mitosis is the process of cell division in the regular cell, or any cell that is not a sex cell. And mitosis occurs in basically 5 steps. It starts with interphase, where the genetic material of the cell is copied, and so there's 2 sets of the genetic material. Then you have prophase, where the DNA condenses to become chromosomes. That's prophase. After prophase comes metaphase, where all these chromosomes, now known as sister chromatids, line up in the center of the cell. So you have the center of the cell here, they're lined up like here. So it's like that. You can imagine. They line up at the center of the cell, and that's metaphase. Anaphase, the next step, is when cells, the sister chromatids in the middle, split apart to one side of the cell. And in telophase and cytokinesis, the last steps, the organelles and the nucleuses begin to form in the 2 cells. In the one now big cell, one giant cell, everything is. There's 2 of everything, 2 of every mitochondria that there was before. Now there's 2 nucleuses, 2 rough ERs, smooth ERs. They're all moving to one side of the cell, and then the cell cleaves in the middle like this and splits apart into 2 cells. And that is mitosis.
You think this is the best? Hey, we're back, and we'll be talking about meiosis next with these beads. We're going to be explaining the process of meiosis by filming the process of Emad moving them back and forth. So yeah.
Hey, Professor Phillip.
Hey, Professor Emad.
Ready to explain meiosis?
Yes.
Alright, let's go.
Okay, so in meiosis, during interphase, like Professor Emad explained, the genetic information is synthesized. Now, this is prophase 1, where these are sister chromatids and they're all in one cell. Now, during metaphase 1, the sister chromatids go to the center, they line up in the center, and then they're split apart. That's anaphase. And when a cleavage finally forms, it becomes one cell, two cell. That's telophase. Now, we go on to prophase 2, where there are two separate cells. They go into metaphase, where they line up, and then they each split to opposite sides.
Now imagine that this paper is some sort of line, okay?
So line here,
so this is
line
one
here.
cell. This is one cell right now.
One cell, two
And-
cell, three cell, four cell.
Yeah, so before these become their own two cells, this would be one cell, this would be another cell. And now they're splitting
Now,
into
when they finally split, this was one cell, two cells, three cells, four cells.
So the
and-
end product of meiosis is what, Professor Phillips?
4 different cells that are all, they all contain the same genetic material.